Hey everyone, is ketogenesis essential for metabolic improvements on a calorie-restricted diet? To address that question, today's guest is Dr. Chung Young Ye, who will present data from his recently published study, followed by Q&A. So with that, welcome Young, and let's get into the presentation.
Hello, everybody. My name's Young. I'm a postdoc from Madison, Wisconsin at the moment. And I'm very happy today to present my work, a recent published paper on caloric restriction and ketogenesis. So allow me to get situated. So hope you can see the slides here. So this paper was recently published in Aging Cell, and we're looking at the effect of ketogenesis in mice that are caloric restricted. So I want to start by a little bit more background information. I think for your audience, I'm sure everybody knows that food is a big part of our healthy aging process, as well as your metabolism and how your body reacts. So even in antiquity, um, people know this. For example, Hippocrates has said that let food be thy medicine and medicine be thy food. And of course, food is the fuel for our body, and there is a lot to know and understand. So I want to give a little bit of overview of all the different types of dietary interventions. So calorie restriction is considered to be one of the most original and dietary interventions. And since then, we've been working on trying to understand how this intervention worked to improve lifespan, metabolic health, and also delay diseases. And that has given rise to many other kinds of dietary interventions that are derivatives. So this include protein restriction, essential amino acid restriction, and that sort of give rise to its own subgenres of dietary interventions. So those are manipulation of single amino acids such as methionine, glycine, and our lab, the Lamming lab at Madison, Wisconsin, is really focused on branched amino acids. So we're sort of working at two ends of the historical perspective here. So calorie restriction, the modern idea of it really got kickstarted by this researcher called Clive McKay. In 1935, he published a manuscript showing this lifespan extension effect in male rats. So this was sort of reorganized here in the lifespan graph. You can see the athlete animals, which are the animals that are free-fed, are the mice, are the rats that are shown in circles here. And both the square and the triangle are rats that are caloric restricted. You can see that they have this pretty dramatic effect in their lifespan extension. And since then, we've been trying to figure out how this effect takes place. So I sort of mentioned a couple terms here that I just want to clarify, and that is ad libitum or ad lib. And this is what is described as free feeding. So as often as you want to eat, as much as you want to eat. So all of us people here, you're ad lib. And calorie restricted, it's a form, a derivative of ad lib. So from the ad lib condition, you're reducing your total energy intake about 20 to 40% without malnutrition. So typically, a lot of these experiments will be supplementing with vitamins or micronutrients. And one of the experimental challenges here that I want to get across is that you're restricting from the ad libitum condition. And this is sort of very difficult to conceptualize from a person's perspective. If you want to run a very well-controlled experiment, is sort of a challenge. So I want to sort of illustrate this. So typically, if you are ad lib, let's say you're eating 100% and you're restricting 30% of your energy intake. So that's fine. So you're energy restricted by 30%. But what are you going to do the week after that, right? So you no longer have a baseline of energy restriction. And as we know, there's going to be a lot of different factors from your everyday life, such as your aging, your different activity levels on the day. And for people, they're going maybe get sick and maybe the weather changes. So that's going to change your energy consumption, your energy expenditure. how are you going to accommodate for that? So that is one of the main difficulties associated with humans carrying out calorie restriction. Experimentally, we try to get around this by pair feeding. So I want to show you guys here. So you have two group of mice, and one of them will be ad libitum the whole time. They're eating as much as they want. And we use that information to inform our calorie restricted group how much they will be eating. So this CR group or the calorie restricted group are, so to say, yoked to the ad lib group. And that because they're living in the same room, in the same conditions, we can sort of have a baseline to work off of. So all the experiments I'll be showing you today are calorie restricted mice formed by this model of restriction. Okay, so another thing that arises because of this experimental design is binge eating and fasting. So if you're only given one meal a day, which many of these animals are because it's very difficult to get very tiny meals to these animals. So they will binge eat all their food within 3 hours. And what happens is that they don't eat for the rest of the day. And of course, they'll go to sleep and do whatever else the mouse does. So they have this long 21-hour period of self-imposed fasting. And we know that there are a lot of benefits for fasting, right? This is typically very beneficial for weight management. You improve a lot of autophagy processes. It's anti-inflammatory, and it's good for your metabolic health. So this sort of gave rise to a different kind of dietary intervention, which I know a lot of you are very familiar with, known as time-restricted feeding. So a lot of people have been questioning these models for calorie restricting, whether or not the fasting component itself has physiological benefits. So this is a very reasonable question. It's a sort of a confounding factor or just as part of the elements of the diet. So these are some of the data that's done by previous graduate student in the lab. And her name is Heidi Pack. She's currently at Dallas at UT Southwestern at the moment as a postdoc. So she tried to answer this question by removing the fasting element of the CR diet, of the calorie-restricted diet. So in the middle here, essentially she made these diets that are diluted with cellulose. So they're 50% energy concentration. And the result was that the animal will eat as much as they want, whenever they want. But they'll end up eating 30% less total energy. And what she found was that these animals are not as healthy as the animals that are calorie restricted the normal way with fasting. So on the left, this is a total frailty graph as mice approach their late life. The black line are the animals that are typically in the calorie restricted experimental design, while the blue are the animals eating the diluted diet. You can see that have higher frailty animals, which means that they have more accumulated deficits. And on the right, you can see that these animals actually live shorter than the animals that are ad lib. So they don't have the benefit of lifespan extension when they lose that fasting component. So if anyone's interested in reading more of this, the paper is published in 21 at Nature Metabolism. We also used a similar model of taking away the fasting, testing the fasting component of the calorie restriction in Alzheimer's disease mouse models. So this is work done by Reggie Baby-Garija when she was a graduate student in the lab. On the right, you can see that the diluted diet actually makes tau phosphorylation worse. So all this is to say that there's a big component of the calorie restriction diet that is within the fasting period itself. So as I mentioned, the animals are fasting throughout the day, and then the fasting component itself is important, but why is it important? So one of the things that people have been suggesting is the ketone production. So ketogenesis during the fasting component. So we know that ketone ester supplements uh, are really good in improving Alzheimer's disease symptoms. Also takes a lot of big part of fatty liver disease. So we need this um, in order to have a healthy physiology. And it has also been shown to extend lifespan, but this is sort of very dependent on when you start the supplement in the animals. So just a little bit of overview on ketogenesis. So there are many different situations your body would go into ketogenesis, and typically ketones are seen as a backup energy currency. So when glucose is low, your body tend to take fat and produce these molecules that are able from the liver that is able to traffic to your peripheral organs and also your brain that needs energy. So there are a lot of sources of energy, sources of ketones, and the sources that can generate ketones, such as your gut bacteria, and also you can get it from exercising as well as supplementation. Okay, so going into a little bit of the biochemistry here. So on the left, it's sort of the source of the ketogenesis. Most of it occurs in the liver. A little bit of it is in your gut and also your kidneys. But the majority of the ketone currencies are coming from your livers. So you're starting with fatty acids that's mobilized from your fat storage, which then go through the beta oxidation process. And you have this molecule called HMGCS2 that's able to convert your initial substrates into ketones. And eventually what you end up with is this molecule called beta-hydroxybutyrate, or BHB. So this is one of three ketones, and BHB is the most prevalent ketone out of the three. So if you see the other green molecules here, acetone, and also acetoacetate, that are precursors also going in other direction as beta-hydroxybutyrate. So those are sort of less than 10% that's in your body of your total ketones. So most of it is going to be BHB. So BHB then circulates through your body and go to your different parts of your body that would need energy at the moment, such as your muscles when you're exercising. And on the right, you can see that through the reverse pathway, BHB gets converted back to acetyl-CoA, which then goes into the citric acid cycle. So this is a way for the body to traffic energy by converting into different energy currencies. All right, so the main question of the paper that we're going to start discussing is whether or not this ketogenesis is why fasting is important for calorie restriction. So that's the central hypothesis here, is that the ketogenesis is needed in the CR models with fasting component. Okay, so this is the experiment. So what we've done is that we made this animal with whole body knockout of the enzyme that I mentioned a little bit earlier. So these animals are HMGCS2 knockouts. And then we take their litter mates, which are wild types, and we put them on either the ad libitum diet. And from those animals, we're able to inform the baseline of food intake. And then we put a separate group of animals that are at the same time on the calorie-restricted diet. So this is just a Western blot confirmation of what it looks like when the animals are knocked out of the HMGCS protein. Obviously, there's no protein detected whatsoever in knockout animals. So these animals will go through a step-down protocol. So we don't actually go directly from 100 to 70. So we go down 10% every week until we reach 70. And then the animal is kept on this diet for 2 months before we started doing a lot of testing, which I'll be showing you guys. And the testing mostly involved metabolic challenges. So for example, glucose tolerance test, insulin tolerance test, which will examine the glycemic control of these animals. I put the mouse in metabolic chambers, which is an indirect way of looking at energy expenditure. And finally, I do have a little bit of molecular physiology data as well. So so all the colors, it's going to remain the same throughout the rest of my data. And I'm going to go between males and females just because some of them are a little bit more straightforward. But essentially, there are not that much different sex effects between males and females. So the important part is to see the blue group and also the purple group. So the blue group is the wild type mice that are fed on the calorie-restricted diet. And the purple group are also fed the calorie-restricted diet, but they're knockouts. So right here is the body weight of the animals over 7 months. And you can see that there is not much difference between the blue and the purple, showing that the loss of ketogenesis don't affect the body weight of these animals. And you can see when you compare the black and the blue, that the animals that are fed the calorie-restricted diet typically don't gain as much weight over time, which makes a lot of sense because you're taking in less energy. So this difference is also observed in the body composition of these animals. So this is the fat and the lean mass of these mice. So as the mouse do not gain as much weight when they're on the calorie-restricted diet, they also have less both fat and lean mass. And if you express that as a percentage, on the right, you can see the percent body composition of these animals that they're leaner as a result. And this is the beginning of the glucose tolerance test, metabolic tolerance test. So they also do this in people using oral glucose tolerance tests. But in this example, we inject the mice with a bolus of glucose. And we measure their blood glucose over time. So what will happen is that the animal will have a spike in their blood glucose, and we measure how fast these animals are able to bring it back down to baseline. So you can see in the left graph here, over time, the animal have this spike of glucose, and animals that are fed the calorie-restricted diet have this enhanced ability to both prevent the maximum peak from going so high, as well as a faster rate of bringing it back down to baseline. And this is not affected by the genotype. A pyruvate tolerance test, it's a very similar test as a glucose tolerance test, but at the same time, we're measuring the rate of the animal being able to convert pyruvate into glucose. So if you are familiar with exercise physiology, lactate is produced, and this is an intermediate between lactate and glucose. So you can see the result looks very similar to the glucose tolerance test, where the animals on the calorie-restricted diet, it's able to lower their blood glucose rapidly after the injection of pyruvate. Okay, so I'll show you this, I guess, the time graph a little bit earlier. When you feed the animal, they begin to fast, right? So the two tolerance tests I showed you earlier would be, the results will remain the same. Doesn't matter what time you do the experiments. However, for insulin, it's a little bit different story. So we test this at two different time points. If you test this at 21-hour fast, which means that the last time these mice were fed was the day before. And right before you feed them again, you test their insulin tolerance. Or at 7-hour fast, which is right before the dark cycle, you perform this test, in insulin test. So here are the data of the animals that are tested for insulin tolerance at 21 hours. So you can see, first of all, the blue and the purple, much, much lower than the black, the gray, and the red, meaning that these animals are much more responsive to insulin. So with an injection of insulin, their blood glucose goes down much further. But if the animals are knockout, you can see that they're also a little bit more sensitive compared to the wild type animals. So very interestingly, if you test this at 7 hours, this relationship is reversed. So the knockout also improved the insulin tolerance a little bit, but the animals compared to the ad libitum fed, calorie restriction tends to make the animal very insulin resistant when you test it at 7 hours. So this is a very time dependent phenomenon. It's actually very interesting. A lot of our physiology not only locked to the circadian rhythm, which is related to light, it's also locked to when you eat. So this is a little bit of metabolic rhythm. So this is a lot more characterization of this phenomenon. It's also done by Heidi, and this was published in 24 in Cell Reports. Okay, so looking at the metabolic chamber data, this is a way for us to look at the energy expenditure of these animals. And this is a time graph over 24 hours. Remember, we're feeding the mice around 6 a.m. You can see that there is a huge peak in energy expenditure of these animals as they begin to eat and digest the food. So they have a huge spike of energy. But there is not too much difference between the blue and the purple, which means that the knockout does not have that much of an effect. And you can quantify this by looking at the area, the average of this over either the dark or the light period. You can see that the animals that are calorie restricted, which are the blue and the purple ones, are a little bit lower than the ad libitum fed animal, which makes sense because they are consuming less energy, so they have less energy to expend. So the respiratory exchange ratio is a little bit more interesting and complicated. So this is a measurement of how much animal is using carbohydrate versus how much they're using fat as an energy source. So the higher the number, closer to 1, it's going to be more closer to pure carbohydrate usage, whereas closer to 0.6, closer to fat usage. So you can see a clear separation between the knockout mice, which are the purple on CR, and the wild type mice, which are the blue on CR. So they have a higher respiratory exchange ratio, which means that they tend to favor carbohydrate as an energy source, which is very interesting because you remember that we did stop the ability of these animals to burn fat as much. But when you look at the females, they sort of have this opposite reaction here. You can see the blue line, especially right after feeding, have a much higher favorability for burning carbohydrates, whereas the knockout mice do not have this as high of a peak. So this is something we're sort of, the sex difference here is something we're sort of trying to figure out in the future. Okay, so moving on, looking at subcutaneous fat of these animals. So you can look at the adipocytes and quantify their weight, their size, and overall, you can see in the CR column, the calorie-restricted column, they look much smaller than the ones on the left, which are all the ad libitum ones. We don't see too much of an effect of the genotype. Some mice tend to get a little bit fatter on the knockout, but this effect is not as consistent as and robust as I've seen. Okay, looking at a little bit of molecular physiology, we look at the autophagy pathways in the liver. You can see that P62 and also LC3 are a little bit elevated in calorie-restricted animals. So P62 is sort of an adapter between LC3 and also the lysosomes and the polyubiquitinated proteins. So they work in the same pathway to recycle proteins. And this is highly elevated in the CR animals, but we don't see an effect of the knockout. So we sort of expected to see more of the contribution of ketogenesis, but that is not the case here. All right, so I sort of did not touch upon ketone itself yet. And these are the ketone level of these animals. So these are ad libitum animals. I measure it 2 ways, using a ketometer that you can buy off Amazon, and also use a colorimetric methods. So you can see in both of these graphs, the wild type animals after 16-hour fast have very high level ketone, beta-hydroxybutyrate levels specifically. And this does not happen in the knockout animals. And this could happen, trying to figure out the molecular pathway here. This could happen in many ways, right? In the flux perspective, because I'm measuring a static measurement of the ketone level. It could be that they are being consumed very fast or they're not being produced, which is sort of what we want to confirm. And if you look at HMGCS2 here, the substrate coming in, it's fatty acids, and what's going out of it is BHB. So I may design these experiments to challenge the animal to confirm that whether or not they're able to convert the fatty acids and also burn beta-hydroxybutyrate. So these are very similar to glucose tolerance tests, but now we're measuring ketones. So everything on the y-axis is beta-hydroxybutyrate. You can see in the blue and the black that animals that are sort of given this lipid challenge, essentially salad dressing or olive oil, and they're able to, over time, over 4 hours, generate a high level of beta-hydroxybutyrate and engage the ketogenesis. And in the CR animals, which are the blue ones, they're able to go much higher compared to the ad libitum fed one. And in the ketone tolerance test, where I straight up give them the BHB, you can see that in the CR animals, which are the blue and the purple, they're able to bring it back down to baseline a little bit quicker. So what I'm showing you is that, number one, I confirm that these animals are not able to engage ketogenesis when they're given straight triglycerides. But if they do have the ketones, they're able to burn it, and very quickly as well. Right. So I did show you a lot of these data, but remember, this data I'm showing you are all ad libitum mice. So what I was interested in was in the calorie-restricted animals. And this is sort of the crux of this paper. So it's going to feel a little bit like I'm tricking you, but I was tricked, and that sort of took me a long time to figure out this story. So this is the experiment. I just want to see what the ketone level of the animals are throughout the day. So I spend a day with animals, and every 4 hours I take their blood ketone. So this is your typical experimental condition. The ad libitum mice, there's nothing happening to them. They just have their food all the time, they have their water all the time, and every 4 hours I measure the ketone. The CR animals I fed at 6 a.m., just like every day, and I measure the ketone every 4 hours as well. So this is what it looks like. So this is male and females together. You can see that the animals that are knockouts have very low but fluctuating level of beta-hydroxybutyrate. So that's the red and the purple. And the black, it's ad libitum animals. So you can see that it does go up a little bit when doing the dark cycle, but the CR animals are a little bit higher. So everything seems to as we expected. But when I take the ad libitum mice, which I did just show you, and I fast them, I perform the same 24-hour study, you can see that when you enter the dark cycle, that the beta-hydroxybutyrate increases, right? These animals are engaging ketogenesis as you would expect, as they are being fasted the whole time. So there's no CR mice here because they are fasted all the time. So what's very interesting is these two graphs I just showed you. And what I had to realize was that when you put them on the same scale, it looks like this, right? So everything in the first graph on the typical day, the ketone level these mice reach are very low, even though there are the fluctuations that you sort of expected. It doesn't really reach the fasting-induced ketogenesis that you're seeing from the ad lib animals. So the question became, why does the CR mice, which is fasting in the first condition, why do they not engage ketogenesis? So that was very shocking to me, and I had to figure it out. So we had to go back to a separate group of animal And I essentially test their fasting-induced ketogenesis every week as they start the experiment. Remember, I told you there was first 8-week onboarding period for these animals. So I would measure that every week. So the first week doesn't count because they're eating 100%. So they're essentially out. But then after that, I would take their blood pH levels every week. So what I found here is that, so we're going to ignore the knockout mice here. So if you look at the black, that's the ad-lib animals. So every week I measure their fasting blood BHB level. You can see it sort of fluctuates, but pretty predictably it goes up. Yeah, and so they're being fasted once a week. And the CR animals, the blue line, they have fasted every day, and I measure their blood BHB level. You can see essentially after the first month, they start to not respond to the fasting, and the blood BHB level goes down to the same level as the knockout animals. So I was able to sort of quantify the area under the curve. So what this shows you is that over time, with repeated fasting, animal have sort of this physiological adaptation to not uh, keto engage the ketogenesis in response. And so this is sort of the summary graph of this finding. Okay, so sort of in summary, we show that we could get rid of fasting-induced ketogenesis, but what was really interesting was that the model itself, that our presumption that the fasting would induce ketogenesis is not true as the animal physiology adapts to fasting. So this was sort of a very long-held assumption in the field. And everybody always said that somebody has to test this. Eventually, it sort of was proven false. But like the premise was false, not that the hypothesis, right? Okay, so I want to show a little bit of human data. So this is a study that was done in the UK in 2011. So they have these women that were being placed on negative 25% restriction over 6 months. And there's two groups. So there is the intermittent energy restriction group. So every 2 days out of a week, they would have pretty severe restriction. And then there's the continuous one. So this is more our mice model where every day they have pretty mild restriction. And what you're looking at down here, you pull out of these pretty huge human study parameters is that the ketone levels. And you can see it on the left, you have people with their baseline ketone level about the same between the two groups. But 1 month, 3 months, 6 months in this intermittent, the 2 days a week, severe restriction group, they have this pretty robust increase in the ketone levels. But if you look at the continuous energy restriction level, you can see that by 6 months, essentially it's baseline. So they are used to the fasting and they no longer engage ketogenesis. So this is to show that what I'm observing is pretty specific to the protocol we're using. So if you have less frequent fasting, you would presumably have more of a ketogenic response. Okay, and a little bit of shortcoming with the data I've shown you is that there are a lot of different types of caloric restriction models as shown here, and they all have different variety of effects. So as I mentioned, we feed the animal in the morning every day, so that animals are active in the nighttime, so presumably it'd be better to feed them in the beginning of the dark cycle instead. You can feed them smaller meals throughout the day, and that also has a different effect on their physiology. So other people have shown that if you align the circadian rhythm with your metabolic rhythm, you tend to get bigger benefits, although they all have benefits, just different sizes of increments. All right, that is sort of what I want to share. And if people are more interested in more calorie restriction studies at UW Medicine, Ricky Coleman, and also Ross They work on the pretty famous rhesus macaque studies where they showed that it hold back age-related diseases. And there are a lot of other people working on the feed timing related to calorie restriction. So as I mentioned, Takahashi lab at UT Southwestern, and also PennDOT at the Salk. And there are a lot of other ways to get ketones. So ketone supplementation is something John Newman and Brianna Stubbs are working on at the Buck Institute. Okay, so I'd like to acknowledge the lab for all the help they have, especially my students who are working on this project, the Wisconsin ADRC, and also the NIA for the funding that they've given to support some of this work. All right, thank you.
As always, I have questions. So I started out with obviously when I prepare, for interviews and scientists to present their data, I always start off with a list, right? So stuff that I want to ask first. But interestingly, because basically the one of the main stories of your paper, of your great study, was that the calorie-restricted animals, produce lower levels, or shouldn't say produce, that there are lower levels of beta-hydroxybutyrate in blood, even with that long prolonged fasting window relative to ad lib. So to me, that has now emerged as my top question. So I get that initially it looks like maybe calorie-restricted mice are producing less ketones. In effect, that's ablated when you knock out the BHB production. But when you consider, have you considered mitochondrial dysfunction as a reason for why the ad lib mice have higher circulating levels at any time point? And in support of that, it's well documented that calorie-restricted mice have better mitochondrial function relative to age-matched ad lib. And beta-hydroxybutyrate is indeed used as fuel for mitochondrial TCA. So could it be possible that it's, calorie-restricted mice should have a big bomb up in production for BHB, especially with that fasting window and being calorie restricted, and that it's less, it suggests to me that it's produced probably at a high amount, and then it's oxidized, right, for energy, leading to lower levels in the blood at any time point relative to ad lib. So what are your thoughts on that mechanism?
Yeah, so that's very interesting. I did show that the CR animals both are able to produce and also consume faster. So that's yeah, so you are correct that it's going to be a separate study. Although, um, a little bit of motivation for this study is to understand what level of ketones that's being reached. So aside from the energetics standpoint, because we're feeding these mice, we're not going to let them starve, they're just eating less. Is that ketones have a lot of signaling properties. So we wanted to understand whether or not they reach a higher level of ketones, of BHBs, that would then go and do protein modifications that would then lead to more signaling properties that are beneficial. So from that standpoint, I think our studies show that it doesn't. But from what you're saying, yes, that would require a separate study to look at whether or not the concentration of ketones within the cell is up like at that micro level, do reach higher or lower, just like transiently. So that's going to be a little bit harder to do, but I think what people can do is to figure out the protein modifications, the signaling property of BHB itself, like what concentration you actually need to achieve different things. Then, as a surrogate, look at in the cell whether or not those are happening, because whatever is happening, it's not observable in circulation. So that is sort of so that would be a challenge for me because I'm working at a different scale, right? And for somebody who is more of a cell biologist and looking at that organelle level, that would be the big question.
Yeah, those are definitely good ways to address that. I have another one. So
Mhm.
what do you think about looking at beta-hydroxybutyryl carnitine, right? So if there's
Mm.
mitochondrial dysfunction, you'd expect that levels of the corresponding acylcarnitine, BHB carnitine, would be higher in the ad lib mice and lower in the calorie-restricted mice. And if BHB carnitine is higher in the ad lib mice, that would add support that mitochondrial dysfunction is potentially contributing because you can't oxidize it's not getting into the mitochondria, right? So,
Hmm.
and it's well documented that acylcarnitines are increased in the setting of mitochondrial dysfunction. So if you measure plasma levels of BHB carnitine, I'd imagine the ad lib mice and the fast, over the time throughout the day would have higher levels of BHB carnitine, and the calorie restricted mice would have lower levels. If that's true, that could be an easy way to say or suggest that um, it's better mitochondrial function and greater uptake of BHB with
Hmm.
less BHB carnitine being formed as an index of mitochondrial dysfunction. What do you think?
I think yeah. So we do work with Judy Simcox, who works a lot with acylcarnitine and the pathways for energy expenditure coming out of the fat depots. So there is, yeah, I hear what you're saying. But what are you trying to get to in the next step? I think it's what I'm thinking about because we do know that the mitochondria are better in the CR animals. Um, I guess you just want to understand how the BHP carnitine it's part of that, right? So
Yeah. And
yeah,
if it's
so
lower in the CR mice, that would
mhm,
suggest that uh, there's better the be, it would suggest better mitochondrial function is helping to reduce plasma levels of BHB because you're pulling
mhm.
it out of the blood and using it for energy. Um,
Yeah, yeah, thank you. Yeah, that's a good experiment to do. We do have the samples, so I send it to Judy. Thank
Nice.
you.
But it does get to the question. I mean, this is what's fascinating about your study is it does get to the question of, is there maybe a U-shaped curve? I mean, granted, most people aren't looking at acylcarnitines unless they're doing plasma metabolomics, and I'm actually going to go look. I have metabolomic data of my own 22 tests, and I haven't looked at BHB carnitine levels over time, but I'm going to look that up. But um, it does get to the question, or your study raises the question of, could there be a U-shaped curve where, so if you don't fast, you're not going to have, if you're chronically fed, at least in the initial state, you're going to have negligible or probably undetectable levels of BHB. And then, the bottom of the U would be optimal. And then maybe too much, as in the ad lib mice, clearly ad lib mice live shorter than calorie restricted mice. Too much may be bad. And this kind of ties into this whole, I don't want to say keto craze because it gets to a dose makes the dose is what's most important. Where if people are like saying, "Hey, my ketones," and they have ketone meters, and some people are some actually MDs are saying your ketone levels are greater than 2 millimolar or some outrageously high number, you're an autophagy. Well, your data would actually show it hit. Well, anyway, that's a separate story. But what I'm trying to say is that U-shaped curve. Um, could there be an optimal plasma level that's not only indicative of optimal mitochondrial function, but that it also indicates that you may be getting some of the potential metabolic and other effects related to fasting?
Yeah, definitely. So, as you know, if it gets too high, then you enter ketoacidosis. That could
Right.
be pretty dangerous. And obviously, that's sort of not what you're saying. You're saying like before that, as far as like the physiological benefits in the range of it. So, I think really interesting is that in people, so for first thing is that there are a lot of sources of ketones. So if you notice in my data, even though animals are knocked out, they still have ketone levels, right? So the ketone are coming from different sources, as I mentioned earlier, that they could be coming from your gut microbiome. There's also a documented pathway for making ketones from leucines, but that only accounts for a very little amount. But if the animal were to be starving, they can force themselves to make ketones. So they could be indication of sickness once you get to that point. So I don't know. So in humans, I guess the second point I wanted to mention is that it's actually very hard to be fasting. So these animals are mice, the data I'm showing. So in one day, they're able to reach like fasting ketone. What I realized that if you eat one meal a day as human, I don't think your ketone level goes above like one, like one or two maybe. It doesn't go up that high. Our metabolisms are just much slower. The fluctuation most people are looking at like 0.6, 1, Like if I was just sitting here and I just measure my ketone like every hour with while eating food or and not fasting, it would fluctuate in that area. But I'm not going to get that sort of ramp up that I showed you. So I think people are talking about different things as if they're the same uh, science. Um, I think it's very important to separate what you're trying to achieve. Do you want a high level of ketone or do you want to be fasting? So that's sort of uh, the point I want to make is that people fasting for ketone is maybe not the best idea. So I try to, because I know people who are of Muslim faith, they practice fasting. And I realize, and I think obviously I'm a scientist, so sometimes I do stuff myself. And like I tried to do that and to see if my ketone would go up, and it just doesn't. Like it's like I fast all day, but my ketone are still the same level. And typically people will eat a big meal in the morning as well. Um, so it's not the ketone that's involved in like the benefit people are searching for, I feel. And if they are benefits, which they are, maybe supplementation would be a better or more effective way if you don't want to fast for over a day, say.
Yeah, definitely. So along those lines, maybe the magnitude and getting back to that potential U-shaped curve for what may be optimal for plasma levels of BHB as a marker of fasting slash ketosis. So you cited, I mean, the calorie study in your study, but so
Mhm,
I went and dug into the calorie data, and as you mentioned in the paper, they saw a significant increase for BHB levels. And granted, that was in people that were 12% CR, but the
mhm,
magnitude of the increase was only from 130 millimolar to 150 millimolar. It wasn't anything crazy, right?
mhm,
So now nobody knows if CR in people is going to do anything long term, but when you consider the positive metabolic effects of CR, I'm not saying the BHB is involved, especially based on your data, which suggests that BHB is not involved in the positive metabolic effects of CR. But maybe the magnitude of the increase is not 0.15 to zero point 0.6 and 1 and whatever else, but a small but significant increase. Now, then the big question is that clinically relevant to actually do anything beyond metabolic health? And if not,
mhm.
are there other mechanisms that BHB can impact that we just haven't looked into yet?
Yeah. So again, in the calorie study, even though on average there's that significant increase, but the people that actually enter ketosis are actually quite low, like it's like less than 5%. And the change is just it's not consistent if you think the diet is doing, right? It's not consistent with the story. Because if your CR, it's supposed to get you put in ketosis, then everybody would have it. It would be like 100%, but that's not the case. And as you mentioned, the change is very small, but um, I just want to say that that is the circulating level. And it's possible that inside the cells are still high or they're high enough to do their benefits. That would be that like remains the question that we try to answer, right? Like at the molecular level, is it enough? Even though we don't see it in circulation.
That gets me thinking I need to look up beta-hydroxybutyryl carnitine levels in CR versus in people versus controls,
Mhm,
metabolic. If that's lower in the people, that could explain. But even if BHB carnitine is lower in CR people versus ad lib, um, it doesn't address, what is an optimal level. If we assume that fasting is going to be good for quote unquote health, but maybe not
mhm.
metabolic health, what mechanisms could be impacted? I mean, you mentioned in cells, I mean, is this a BHB gets to the brain and then that's the positive effect during aging, or what would you speculate?
It does. So John Newman and Eric Burden from the Bach Institute. They've been working on different post-translational modifications. So BHB does directly, as you sort of mentioned with the acetylcarnitine, it does directly modify HDACs, and it does modulate gene expression that way. But we don't know what concentration is needed to do that, and that's sort of my question. So it does elicit a lot of its effect through its ability to regulate gene translation by modifying the machineries that does that directly. So that's very interesting, but there's a lot of work have to be figured out in that department.
So are you planning on looking at other systems that could be impacted by BHB during aging, including brain, whether it's HDAC or epigenetic changes or immune, to see how that looks differently in the knockout animals versus wild type?
But the knockout animals, um, by themselves, they're not that healthy. So we're trying to move away and go towards a supplementation approach, and also ketogenic diet approach. Ketogenic diets are also not like the best thing because it's mostly fat. So to ketone ester supplementation will be very interesting. I'm starting to move towards studying Alzheimer's disease model. So it'll be very interesting to see how that affects the various Alzheimer's disease models as far as their brain cognition and their whole body physiology. So ketone esters are very interesting. So this is work with John Newman and Brianna Stubbs because in the experiment, obviously they're mice and you would give them hydroxybutyrate salt, and giving salt to patients typically not a very good idea. So they are trying to make these not salt, so they're esters that you're able to take just like food, and that will raise your ketone level that way.
You mentioned that the knockout animals were sick or not very healthy, but had
Uh, yeah,
metabolic health on
yeah.
CR, right? So what was the phenotype of the knockout animals that they weren't generally healthy?
So not in my experiment, since I didn't do like a lifespan experiment, I don't see too much an effect. But for people who are specifically evaluating longevity and also liver health, what they'll see is that if you fast the animals, so what happens when you're fasting, your body, your fat depots try to send, because you're running out of glucose, so you need ketones. So your fat depots are sending fat to your liver for your liver to make ketones out of them. But you can't make ketones. So what happens is you just end up with fatty liver.
Mm.
Um, yeah, so this is actually interesting. Physiology is also very synthetic because you're not supposed to have this deficit, and you get fatty liver, but once you start eating normal food again, the fatty liver will clear out pretty quickly. So this was observed with Eric Creetham at University of Minnesota. So it's like a very reversible effect. And if you just let these animals live, they do have a shorter lifespan. Um, what it is dependent on ketones, but we're not sure. I guess the pathology aspect of why that is. So this is not CR related. I didn't really see an increase in fatty liver in my animals. I presumably they're fasting all the time. They got used to it and they developed other pathways to deal with the fat migration.
But it was a short-term study. I mean, the hypothesis would be they probably, even on CR, if they live long enough, they probably would end up with fatty liver because if you can't,
Possible.
yeah. So that's interesting because BHB levels decline during aging in people. I mean, there are plasma metabolomic studies that have shown this. So is it possible that, and I can't get the enzyme right, is it HGCMS, something like that, too?
HMGCS2.
Yeah. HMGCS. It's like somehow I've turned into dyslexic scientifically, right? But so is it, how does the activity of that gene change during aging in liver? Has anybody looked at that?
I don't know. That's a very interesting question. Yeah, I'll have to look that up. I don't know if anyone has done it, that experiment. This model of mice, I guess we made it a couple years ago, and it's just been put on Jackson's website, which holds all the mouse models. So I think there's going to be more data coming out from different labs that are able to access samples.
Nice. So I have a couple lifespan related questions. So assuming, all right, so with your knockout animals, the metabolic effects on CR were not reduced or impacted with the inability to make BHB. But on a ketogenic diet, and normally, granted, it's, as you mentioned, it's 89% fat. There's the study, they looked at 89% fat, 70% fat, and 15% fat. But it had to be eucaloric, so they had to control calorie intake because, as you know, high-fat diet in mice, they'll just get obese. But on the eucaloric high-fat diet, ketogenic diet, because they saw plasma levels of increase for BHB, you had that extension of lifespan. So, there's this prevailing view almost that, metabolic health is central to health and lifespan related improvements. So would your data add into the hypothesis that maybe not? I mean, there may be other mechanisms that, or at least ketogenic derived metabolic health improvements may not be relevant to lifespan? And if so, which gets me back to that question of, if so, what are the mechanisms on a ketogenic diet, if not BHB and ketosis, that would be relevant for longevity?
Yes, that's an interesting question. So you're right. So one of the shortcomings in my study is that I didn't look at lifespan. And if I get $20,000 and I stay here a couple more years, I can figure that out. But it's so hard to show with our model, because the animal by itself have changes in lifespan. Um, and as far as the ketogenic diets, those are also very difficult to parse because, as you know, animals would get obese. So what they actually do is they cycle the diets. Uh, they cycle the diet. So every week they would go on the ketogenic diet. Eventually they figure out the lifespan effect. So I think the future study is going to be the supplementation. I'm not sure who's working on it at the moment. I believe there are people at USC looking at it. So that'll be a more contained experiment, right? So there's not like the high fat aspect of it. So what's interesting is that I still see a lot of benefits even without ketogenesis, right? In my animals. So without the ketones, I don't know what is lost. I think that's the big question. And again, that's going to go back into changes in their gene expression landscape and how like it becomes not the nutritional aspect of the molecule, but that the cell signaling aspect of the molecule. Yeah, so I don't have like a clear answer because we just don't know at the moment.
No, that's okay. That gets me thinking that so BHB is regulating, gene expression or HDAC inhibition uh,
Mhm.
for, immune-related genes or less neurodegeneration, right? So
Mhm.
I don't know. Has that been measured?
Well, it has been shown, I think it's John Newman's work from, I forgot what year it was, but they do look at brainwave in Alzheimer's disease animals that are supplemented with ketones. And they do sort of have a restorative property in that model. So it can go, so it obviously does go to the brain and it can be like the preferred source of energy. I don't know at that point if they started to look into the gene expression of the neurons yet. So that'll be something to follow up with them.
It sounds good. And then for the ketone salt supplementation study, and as you mentioned, granted, probably shouldn't do it in people, but I'm guessing that's going to be on an ad lib diet, or will you also have a group of CR plus the ketone supplementation?
Well, if we're to do CR, it would be both. So it will always be both. So at this point, I would just be at that ad lib diet. Yes. So it will be on top of the ad lib diet. Usually put it into the food if we can, and see how that affects Alzheimer's disease progression. So that'll be.
So you may have competing things going on, right? Because if on an ad lib diet there's some measure of mitochondrial dysfunction and you give BHB and that can't be oxidized for energy, now you've got a buildup of potentially toxic BHB
Oh,
creatine. And then you've got the other side of it with the higher BHB positively impacting, the brain, so that's an
yeah,
interesting
yeah, yeah.
dichotomy that you could have going on there.
Yeah, that's interesting. That sort of go back to what you were mentioning about the U shape. So like what dose of BHB? So yeah, so once we started doing the experiments, I think one thing to do is to titrate it. So you really need a way to manipulate BHB circulating levels, and I'm not sure at this point how fine of a control we can have on these animals, but if we figure that out, say how much parts per million to put in the food. So then we can sort of answer the question you had earlier, which is like what is U-shaped curve? It's going to look like what is the best dose for what we're trying to achieve. Yeah, so that's a great experiment. I have to write that down. Thank you.
Nice. So, all right, and then so I like I don't want to say inhibiting the fasting window, but the caloric dilution experiments that
Mm,
Dudley. I mean, granted, it's Heidi's work. It's not your work, but because you presented it, that's the only reason I bring it up. So, I mean, cellulose is insoluble fiber. So I wonder, because the shortest lifespan was on the diluted calorie intake, but it's also it's a non-physiological dose of cellulose that the
mhm.
animals would never get, right? Which makes me
Yeah.
think that the intestinal potentially adverse intestinal effects may be worse than interrupting the fasting window there. So,
Mhm.
yeah, what are your thoughts on the cellulose model of breaking up the fasting window?
Yeah, so we do have a lot of criticisms on that model. It's just like an easier way to do it. So obviously what Joe Takahashi has been working on is they have like this machine that automatically dispense food, and they just dispense like very tiny pellets because so what we do is that it's very hard to cut the food for the animals because they don't eat that much every day, and we can only get to maybe point like one or two decimal point accuracy. And but if you have them pre-made, that's a different story. And if you have a machine that's able to dispense the food for the animals, then you can take out the fasting by feeding them. So how do you also have done this is to have multiple feedings a day to try to eliminate the fasting that way. So you would feed one, two, or three times a day. And with the machine, which presumably she's working on now, you can feed them six times a day, and you can feed them throughout the night. So you don't have to be there to feed the animals. So yeah, you're right that it's a more straightforward and better model. But again, that does change sort of how you interpret the data because you don't have the same amount of food, right? And we also know that that is a big contributing factor of physiology. So your gut will sense that there is food in the stomach, and that is it's going to release your your intestinal hormones, and that does have a lot of effect, as we know now with Ozempic and all the weight loss drugs. So, there are downsides to the cellulose dilution model, and there's also, strengths to it, in that you do model the volume of the food itself. So,
Yeah, the machine dispenser, I think, would do a lot of labs, so a lot of good.
mhm.
So another model is the glucagon receptor knockout mice, which you'd expect to have lower levels. They didn't measure it in this study, but you'd expect them to have lower plasma levels of BHB because if the receptor's knocked out, you can't have glucagon signaling. So in that study, they had calorie restricted mice and glucagon receptor knockout calorie restricted mice. And median lifespan was significantly shorter for the glucagon receptor knockout mice. And the metabolic effects, I mean, it's almost counter to the data you saw. Whatever positive metabolic effects they saw in CR, they didn't see it in the glucagon receptor mice, not knockout mice. So it raises the question of glucagon receptor knockout mice should have lower levels of BHB, and whatever other glucagon receptor, whatever other signaling that it inhibited, not the ketone production, um, had something to do with metabolic health. So I know I'm asking you to speculate a lot today, but what
Mm.
would you speculate within the glucagon receptor signaling pathway besides BHB production that could be involved in sustaining metabolic health on a CR diet?
So that's very interesting. I'm not familiar with the glucagon
Yeah, I can send it over.
paper. Yeah, you could send that to me, and I'll take a look at it. So what I'll say is what's very interesting is that the CR animals eventually what uh, they'll do is that they upregulate glycogen metabolism, so glycogen storage. So I think that might play into a little bit of that. So I don't know if you knock out it's just the receptor knockout whole body. So yeah,
I
that's
think they did it both ways, where his whole body
okay.
and
Okay.
liver. I have to look it up again for sure, but
Yeah.
they did it both ways.
Yeah. So I don't know which system comes first. So that's very interesting. Yeah, I'll have to look more into that. Sorry, I'm not too
Oh,
familiar
no
with
worries,
that
no worries.
literature.
I thought I assumed you knew. I should have probably thrown these at you beforehand, but
No,
I don't
no
know if you
worries.
expected this level of questions, right? I have, my background is similar to what you guys did, so anyway, but yeah, if you had to speculate, right? So without glucagon signaling, you should expect to have ketogenesis also impaired. I mean, I don't know how you could have ketogenesis without glucagon signaling, right? So
Hmm.
it suggests that there's other stuff, and there's a lot of things that glucagon signals besides ketone production, maybe upstream, maybe some of the upstream stuff, and I don't know what that would be, but you'd most definitely be more well versed in glucagon signaling and the intermediate steps before getting to ketogenesis. But maybe those are a part of the metabolic health story that's wiped out, and not necessarily BHB. I don't know. Just if you had to speculate, which would it be? I mean, upstream of the ketone body production that could impact metabolic health.
I, I I would not be surprised if they could be run independent of each other. So I don't know. So yeah, I'm gonna have to look into the study. I don't know if they did like a cell autonomous models to show that. So maybe by the cells by itself without any glucose signaling would be I like all the machinery is there inside the cells. So maybe the cell doesn't need the glucagon signal for the ketogenesis to happen. Yeah, so I think that's sort of like the easiest interpretation for it.
I thought they're dependent on each other, because with glucagon signaling, especially on CR, how can you have
Mhm.
ketone body production if you can't signal through glucagon to break down fatty acids to produce BHB, right?
Oh, so you're looking at in the adipose perspective.
So
So what if you have fat already in the liver,
yep.
that then you wouldn't need it.
But you wouldn't have glucagon signaling, right? If they knocked out the receptor, whatever the signal is. So hypoglycemia would be part of the signal for glucagon to signal to produce ketones, right? Um, so if you've got CR animals that are CR and fasted, that 21-hour fast,
Hmm.
they should reach some level of hypoglycemia to impact higher levels of glucagon, and then the receptor's knocked out. So now you can't have ketone body production. And when they used that model, they saw all of the metabolic effects. I mean, lower triglycerides, lower cholesterol, all of that being reduced by CR went away when knocking out the glucagon receptor. So if it's not related, your data would suggest that it's not related to BHB, that BHB is not
Hmm.
that process. But then it makes you wonder, and I don't know what the answer is. It suggests that there's something in the glucagon signaling pathway that can impact metabolic health that is unrelated to BHB. It's just, I don't know which part of that pathway would be involved.
Okay.
What would you think? I mean, would you have any ideas?
Well, a lot of work has been looking at different types of futile cycling in the fat depots. So
Hmm.
maybe just the mobilization itself plays a big part, and you don't need it to to engage the ketogenic pathway, right? So maybe there's a different way for the energy production to happen, or for the energy production to leak out. So I have to look into sort of what the phenotypes that I'm looking at. But I think that's where I would suspect. And also there's a lot of different breakdowns between different fat depots that people are looking at. So there are different roles that would be involved if you were looking at the brown fat or the subcutaneous fat. And of course, what happens to the fat in their knockout model will be very interesting.
Yeah. All right. So you think it's a fat story. All right. So I don't know what the answer is. I mean,
Mhm.
interesting to hear others' thoughts on what it could be. So, all right, future studies, you mentioned you were going to look at Alzheimer's disease, but are there studies you're currently working on and have plans to work on? And then additionally, when considering that you're a postdoc, like
Hmm.
where do you plan on going with this? Like, are you going to stay in ketosis and ketogenesis and calorie restriction? Or like when you've got your own lab coming very soon, I'm guessing,
Mhm.
if maybe you already have that in the works and you've been
Oh,
interviewed, I
not
don't
yet.
know. But when you've got your own lab, what's the Chung Young yeah? What's your research focus? So first the current future studies and then your lab. Where do you see yourself?
Yeah, thank you. So, the project I'm trying to finish at the moment for the Liming Lab is the effect of isoleucine restriction.
Mm.
Also, as I mentioned earlier, there are different derivative, derivative versions of dietary interventions from calorie restriction, and one of them is isoleucine restriction, where we just take one amino acid out of diet, and that does a pretty drastic effect. It does extend lifespan, and I've been working on the molecular mechanisms of it for a while. And one of it is TOR, and I've done a lot of knockout models with knocking out TOR in the liver to show that what its involvement is with the expression of the dietary intervention. So for my own future work that I'm trying to work on at the moment is, sort of related to the U-shape effect that you're sort of talking about. So a lot of times in our experiments, we sort of follow what previous people have done, and we focus on very narrow range of what we know would happen. And that's also true for dietary protein. So very few studies have looked at diets that go up to 60% protein. So at the moment, I'm testing in Alzheimer's disease animals the effect of dietary protein going from a low of 7% and going all the way up to 60%. So that's sort of my independent study at the moment. So 60% protein is actually very interesting. One lab has characterized it maybe 5, 6, 7 years ago now, and they haven't really followed up on it. Mice, they will lose a lot of weight when they're on the 60% protein, which is also true for the animals that are on the 7% protein. So you do have this inverted U shape where animals that are fed sort of like the average range of protein tend to thrive or get fat a little bit easier, whereas the animals that are on the two ends tend to react with more increases in loss in their fat mass and become more lean. So I'm trying to figure out what the Alzheimer's physiology react to different react to this different level of protein concentration.
And then when you've got your own lab.
Yeah, so this is my K99 project. So I will take that as part of my lab. As part of my lab, I want to look at so my central interest is aging and aging-related diseases. So I think my data have shown that nutrient sensors themselves are pretty important mediators of the aging process. And I want to understand how we can separate that from the nutritional content that stimulates these nutrient sensors. So I want to use genetic methods to manipulate different nutrient sensors without changing the diet and see how that affects aging, age-related diseases. So that will be the focus of my lab.
Nice. So wait, I have a couple more questions. So the isoleucine restriction studies, I mean, I think you guys have also done other branched chain amino acid restriction studies,
Mhm.
but I worry that maybe it's an artifact of animals living in a cage. I don't know, because if you're sedentary and you've got too much protein, as you mentioned, the U shape with potentially too
Mhm.
much protein being bad. I'm not saying the standard mouse chow is too high in protein, but maybe if you're sedentary and stuck in a cage and you can't move, maybe their protein levels are too high, their intake levels are too high, and then restricting one of the amino or some of the branched chain amino acids, which are essential. Maybe that's kind of like you're getting closer to an optimal physiological dose for them to have improved health and lifespan. I don't know. I'm just trying to think of a way how it can make sense that restricting one or a few branched chain amino acids would be good for animal longevity, especially when they're essential amino acids, right? So, what are your thoughts?
Yeah, yeah. So that's a good question, and yeah, we've been trying to answer that for 10 years in Delhi's lab, and the problem is that we have so much physiological observation because the effect is so drastic, and the molecular data, you sort of just have to run into it. And I got pretty lucky with my study. And as far as the animals being living in a cage, so we have done exercise experiments. So the animals are, so they live in a cage and then we take them out to exercise. So they're not like free living, but they do burn a little bit of energy while they're pulling weight. It's this experiment. So we tie like this little cart on the tails and then they pull weight along the track. So this was Michaela Murphy. She got married. I forgot her name. So Michaela Murphy's paper that was published in eLife a couple of years ago. So they don't. So the opposite of your question, right? If you take an animal eating normal amount of protein and you exercise them, you will reduce fat accretion, but they don't have the same phenotype. So it's not because the effect that we see with the restriction is so drastic, and we haven't really been able to reproduce it using other methods, which would then prove what you're saying. So we don't see that in these animals at least. So if we put a wheel in the cage so they can exercise whenever they want, I think that will partially answer your question. I do think we have that data. I don't think it did anything. And with the branched-chain amino acids and also the essential amino acid point, what's really interesting is that the restriction of different essential amino acids have very different results. So if you restrict leucine, like essentially nothing happens, and then the animal one day will just die.
Hmm.
So it's gotta be some undiscovered molecular signaling that we're not understanding. Uh, and I know it sounds a little cryptic, but if you see the animals on isoleucine restriction just shred so much weight, while the leucine restricted animals just don't, essentially don't react to it. It's it's pretty incredible what that we discovered for in this regard.
How would you translate the isoleucine restriction into human data? I mean, how would you restrict just that
Mhm.
specific? Because they come in the trio, right? It's most foods are rich in isoleucine, leucine, valine. How are you gonna, I mean, unless you reduce
Yeah,
overall protein intake, but then, uh,
and that's just protein restriction, right?
yeah.
So we do a couple ways to answer that question. The first is that obviously we're trying to figure out the molecular pathway involved, so we can do it molecularly with a pharmaceutical drug. And a second answer, I guess we could talk about it now. Is we definitely do have a partnership with people that were in the genetics department. We're trying to make like GMO soybeans that's able to reduce, uh, sort of change the formulation of a millable product,
Oh,
so
really?
that
Huh.
yeah, so it become more accessible.
So they could market a lower level of isoleucine in their soybeans, and that lowering isoleucine in the diet has been shown to extend lifespan, right?
Well, it's very hard to sell like negative product, but that's what we've been trying to do.
Yeah. And then the 60% protein diet, is that study going to be short term, or are you also going to have longitudinal data?
Don't have money for the lifespan experiment yet, but hopefully. So my project right now is run 4 or 5 months. But I need to collect the tissue to do more molecular studies. I haven't had the chance to do that lifespan experiment, which would be very interesting. Yeah. So
Yeah.
I don't know what will happen. Yeah, I'm not sure.
Well, I would speculate that well, longitudinally, so protein absorption, actually
Mhm.
digestion and absorption declines during aging, and we know that because more protein and amino acids, actually more amino acids reach the colon, the large intestine, and then there's protein fermentation, where those gut bacteria derived protein fermentation derived products are found in the blood, doxyl sulfate, pericresol sulfate, TMAO. So if your study is short term, you might see positive systemic effects, but then I would worry maybe the mice get to a certain age, and then now you've got an increase in these protein fermentation products. So it is one way maybe to get around that. I don't know, it's probably too late. But if you got like a CKD animal model and you use that as kind of like an accelerated aging phenotype where
Mm.
you put that on a 60% protein diet, because you can't age the mice. If you can't age them, how would you reproduce the metabolic phenotype of now you've got more protein reaching the large intestine and now you've got more of these circulating protein gut bacteria derived fermentation products,
Wait,
which,
why can't we age the mice?
oh, I thought you mentioned that, to do it for a few
Oh,
years, too expensive.
I,
But if you can age them, even better, right?
yeah, yeah,
So,
yeah. I can't age the mice because I have no money to age them.
yeah.
Not because the technical limitation.
But
Yeah.
but then is there a way without, having the money to do it right now, where you can get
Mm,
the answer of is that 60% fat diet, um, so where you could speculate, right? Because so if you don't have the
yeah.
longitudinal data, maybe there's a way to reproduce the protein fermentation products and that high protein diet in the short term, because maybe short term you do see indeed favorable metabolic changes, but I mean, even things like high protein diet, knowing that kidney function declines during aging, and it does in mice, there's going to be an increase in blood urea nitrogen over time because the amino group is going to be converted into urea. Urea degrades tight junctions. So now we've got this effect of protein being fermented by gut bacteria. And we've also got another effect of very high protein, more urea production because the kidney can't clear it. And then that's going to degrade tight junctions and potentially lead to leak into the blood and immune activation. All of this, though, being more likely in the aged phenotype, right? So in the short term,
Mhm, mhm,
the kidney function is probably good enough where it can handle the bigger urea load systemically and probably can, clear and excrete the gut bacteria derived protein fermentation products. But maybe you get
mhm,
to a certain age and that just isn't the case, right? So anyway, I'm just wondering if there's maybe a model where you could address in the short term the effect of high protein diet, high urea, high uremic metabolites that are derived from gut bacteria on all of the phenotype, all of the metabolic and other phenotypes that you're looking at, uh, because you may get a good answer in that short term, and then everybody's like, "Hey, let's go to 60% protein diets." But then the longer term effects may be, understudied or not studied, and we've led people astray down the wrong road. So, what do you think?
yeah, I think that's very interesting. You're completely right. Long-term effects are unknown, but maybe in a few years we'll have reports from the people who went on carnivore diets, and we can sort of get a little bit data from that and try to understand what the protein signal is.
It gets tricky though, because most people anecdotally on carnivore diets are physically active. Physical
Hmm.
activity can actually improve short chain fatty acid production by gut bacteria,
Hmm.
which is one part of the signal for reducing growth of these protein fermentation bacteria. So if you have high levels of SCFAs, the protein fermentation bacteria can't grow in an acidic environment. So for people on a high protein diet that are physically active, that could be a mechanism to suppress these protein fermentation products in the short term. But
Hmm.
with kidney function and age-related decline kidney function, I don't know. But
Yeah.
maybe
Yeah, so we did keep track of I try to keep track of kidneys of our animals. So we do have the tissues. I just haven't done the molecular analysis. We weighed all the kidneys, and they don't seem to be that too much changes that we observed yet. So, but maybe I need to do it for longer in this
But I
study.
wouldn't say, I wouldn't suspect, and I wouldn't, because now people are going to yell at me in the comments, a high protein diet doesn't impair kidney function. That's not what I'm saying. I'm just saying the kidney in the young state on a very high
Hmm.
protein diet, it would potentially handle the increased burden of protein fermentation products and urea because it's young. But with the age-related decline for kidney function, which is well documented, that a very high protein diet may be less efficacious and even potentially
Hmm.
deleterious when considering negative systemic effects of urea and the uremic metabolites on health. But then it just gets to the question of how could you evaluate that in the younger state before getting to that 2 year later study because it takes forever to age the animals.
Yeah, that's very interesting. Yeah, I'll keep an eye on that. And we do have studies going on that look at just in older mice that are switched to higher protein concentrations. But I think those studies that are ongoing tops out at 36%. So, but maybe because it's a little bit of a gradient study, so maybe we'll have an effect there if we collect the tissues. Yeah,
Yeah,
thank you.
36% is still pretty high. All right. Okay. So that was great, Young. I appreciate
Yeah,
it.
thank you.
I'm looking forward more to your work in the future. And are there any topics that we didn't cover related to ketogenesis and CR and metabolic health and longevity that you think we should have?
I think we pretty much cover everything.
Cool. All right. Thanks.
Yeah,
Thanks, Young. Have a
thank you.
great day.
It's nice meeting you. Have a good one, Michael.